Welcome students. Under this session, we will be covering a program Bachelor of Science second year, subject Botany, semester 3, course code BOC 103, course title Plant Anatomy and Embryology. Title of the unit, Unit 5, Structural Organization of Flower, module name Development of Male Gametophyte. Myself, Dr. Nisha Kevat, Assistant Professor in Department of Botany from DPM Shri Mallika Arjun and Shri Chetan Manju Desai College. Outline. In this session, we will be covering the male gametophyte, microgametogenesis, in other words, development of male gametophyte. What exactly is the difference between the generative and vegetative cell? Learning outcome. At the end of this presentation, a student will be able to understand the developmental process of the male gametic formation. Male gametophyte. Male gametophyte generation begins with the formation of microspore. Just a brief background, we know that in a flower there is a stamen which is referred as a male reproductive organ, and it is a dithecous and tetrasporangiate. This tetrasporangiate structure which is formed has an individual unit which is called as a microspores. These microspores are released in anther locule by breakdown of a common callose wall of a spore tetrad. Since when they are formed due to the divisions, they are attached as a tetrad, but when they will want to have their individual identity, they need to be broken down and separated out from each other. The microspore after the release from the tetrad will be referred as a pollen grains. Now, pollen grain represents the first cell of the gametophyte. Microgametogenesis or development of male gametophyte. After being released from the spore tetrad, pollen grain absorbs the locular fluid and they will expand considerably. As a result of expansion, vacuoles will be very prominent and the cytoplasm will get limited to the thin film lining the cell wall. We can see here as a picture of the pollen grain. After the locular fluid absorption, it has expanded. As a result, cytoplasm will move towards the periphery. We can see here the nucleus shifts from the center to the periphery and prepares to divide mitotically. Here the nucleus which was there in the center, it has moved to one particular end. Nuclear division is asymmetric and it will give rise to the two unequal cells. The larger one will be referred as a vegetative cells and the smaller one is called as generative cell. In this we see this nucleus undergoes a division mitotically and give rise to the two unequal cells. The larger one is vegetative and the lower one is generative. There was a wall which separates the vegetative from the generative cell. After some time, the partition wall between the two cells disappears and generative nucleus will lie within the cytoplasm of the large vegetative cell, which is clear from this image. The pollen grain thus becomes two-celled. At this position, we call this as a two-celled pollen grain. Now, generative nucleus continues to grow. This is the generative nucleus which continues to grow. The function of a generative cell is to give rise to the male cell, that is the male gametes or a sperm. The generative nucleus divides mitotically, followed by the cytokinesis, and the wall separates the two cells. In this image we see that after enlarging, the cell has divided into two mitotically and both of them have their individuality. Now, these two cells are referred as a male gametes. Once it has fallen onto the stigmatic surface, this pollen grain will start germinating. Here is the pollen tube which has the male gametes and also a tube nucleus. As the time proceeds, the tube cells will go on. As the time proceeds, the pollen tube will enlarge and it will go on increasing in size, and this male gametes will approach closer and closer to the ovule for a fertilization. Coming to the difference between the generative and vegetative cell. Once the pollen mitosis has taken place, their DNA content increases twofold, that is 2 times. Lysine rich histone is associated to the DNA is also seen because we know that histone is a protein which binds to the DNA and in a generative cell their nucleic material is doubling, so they will require more DNA binding proteins. RNA and protein synthesis will be absent and they are metabolically inactive because they are not going to carry out any metabolic pathways or any metabolite synthesis. So, it will be inactive. In contrast to the generative cell, vegetative cells, their DNA content increases to 1-fold. Here the DNA binding protein will be absent because they do not, since their DNA content has not increased, they will not require this histone proteins. RNA and protein synthesis are excessively high because they have to support the generative cell and they will require various types of the proteins. Therefore, obviously they are metabolically active. This is a reference. Thank you.